Okay, thank you for being here. So I decided to speak about this subject not because I am an expert, I'm just essentially starting the last years, but I find the subject fascinating, and I also want to advertise it for more algebraically inclined mathematicians. That is really close to applications. We can say interesting things with more advanced algebraic tools. So, So this is a brief summary. So I will give you first an example and then the definition of what a chemical reaction network is. Then I will define and explain why one is interested in studying multistationarity. And then I will give you some particular examples that are very important of chemical reaction networks with a special algebraic structure, which there are binomials popping up there. And then I will give you some just a rough idea of what can one say with these tools concerning bifunctional enzymes. If I have time, I will say a few words about what's called maximal response, and then I will end. Okay, so this is a very simple chemical reaction network, but which is very important. This was proposed by McKeithen. It's an important paper in the Proceedings of the National Academy of the US in '95. And he was trying to explain somehow there is something they call kinetic proofreading. There is an incredible fidelity in the transmission of DNA. So he was trying to propose a mathematical model for this. And so in fact, so this is the simplest case. Is it working? No, it's not. No. Yeah. Okay, so in fact, this network is a little bit bigger than this, but this is the smallest one. But this is the same. So there are two chemical species. So A is called the T cell receptor. The T cells are the lymphocytes that are responsible for us not having cancer. And it has to, and this is called the antigen histocompatibility complex. And so the question is that this T cell has to distinguish whether it has, the binding is with a foreign antigen or a self-antigen with low affinity. And so what he proposes is that there are two species, they bind, and they produce this species DRC after binding with some rate, which is given by this positive number, and then this can go back. And then this C, before what they say emitting a signal means that this goes and produces another chemical reaction. It has to be first transformed into another complex T that can go back, and maybe it's a sequence of those. Okay? But this is the simplest one. And so how do we build differential equations that explain the dynamics of what is happening? So here we have 4 reactions. The reactions are the arrows. We have 4 arrows, this means 4 reactions. There are 3 complexes, which are A plus B, C, and D. And these are composed by 4 species, which here are just A, B, C, D. And the object we want to study are the concentrations of these species along time. I'm going to call them X sub A up to X sub D. This is what we are interested in. And so this low mass action was proposed in Norway in the 19th century by, to my surprise, but not so much, was proposed by a chemist together with a mathematician, and the chemist is Ragni Piene, great-grandfather. And it's a very mathematical theory. So they first published this in Norwegian, but then nobody knew that they had this idea, and then they republished this in French, but it was only known when they published it in German, which was the lingua franca at that moment, and it had been in the same time rediscovered by a Dutch physicist, Van Hoft. And so what they says they propose that the idea that the rate of a reaction is how this changes with time, is proportional to the probability of collision of the reactants. So the probability of the two molecules that have to bind, the concentration of, depends on the concentration of the two species that have to bind. And with an independence assumption, what happens is that this rate of reaction depends on the product of the concentrations of the molecules that are producing this reaction. So when you translate this mathematically, what we get are just polynomials, and get monomials in this case. So for instance here, this reaction rate is proportional, is this number times the product of the concentration of A and B. So we are, in mathematical notation we write this, but we are going to assign the standard notation. We call this x to the 1, 1, 0, 0, and this is 0, 0, 1, 0, and this is x to the E4. And we are then, in the next transparency, I'm going to call these exponents y's, y1 up to y3. And then what happens is, for each of the species, here A is here, there are 3 arrows coming or arising from a complex which involves A. So there are 3 terms. So this derivative will depend on minus K12, this product, plus K21, this product, but this is just one variable, plus K31, this one, all the same. So here we have, here there are 3 arrows coming, here 3, and here there are just 2, so there are just 2 terms. Okay, but it's a sum of monomials. Of course, we could have 2A, for instance, or for instance, the well-known thing that maybe we all remember from high school is that 2 molecules of oxygen plus 1 hydrogen, plus 1 of oxygen produces water. So here the vector would be 2, 1, 0, 0, 2, 1, 0, okay? There could be some integer exponents, not just 1 or 0. And then in general, what we have is a set of reactions. These will be labeled edges. And the label are positive real constant. If they are 0, we say that the arrow is not there. So if we see the arrow, the constant is assumed to be strictly positive. And then there are M complexes that, sorry, I wanted to point and I did the wrong thing. So, and in the vertices are going to be labeled by these monomials x to the yi, and these are composed by S species. These are in fact the concentrations of the species, but these are the variables. So the chemical reaction network is then a finite directed graph. When we have decorated the vertices with the complexes, this is just, if you wish, as the exponent of these monomials. And the edges are decorated with these positive real numbers, which are the reaction rates. Okay, and we view the concentration as function of time, and the mass action kinetics is the dynamics associated to this thing has this shape. So this is a vector of derivatives. It equals a sum over all reactions, all the edges in this directed graph. Here this is reaction rates, these are real numbers, this is a monomial, and this is a difference of 2 integer vectors, so this is a vector thing. But if you think what is happening, each of the coordinates is numbers, numbers, monomials. So the first observation is what we have is that it's of the form derivative of xk with respect to t equals some polynomial fk in the x variables. So these are autonomous polynomial dynamical systems, ODEs. And just for further use, this can be written as a matrix of numbers involving the kappas and the y's times a vector of monomials. Here this psi of x is the vector recording the x to the yi's, and this is just a real matrix. Okay? And so the first question is, how general are these, the f's that come this way? They are which we call the Hungarian lemma, because it was proved by 2 Hungarian mathematicians, I guess they were mathematicians. They say that the only restriction, if you have a polynomial system of the form d dx equal fk of x, it comes from some chemical reaction network, if and only if the kth polynomial, that was this polynomial here, defining this derivative, has to have this shape. There could be positive and negative terms, but those which are negative need to have an xk. Sorry. So if there is a negative term, there is an XK there. So for instance, the standard this type. Okay, so one assumes that we are going to get something less chaotic if it has. Okay. So what happens is that in general these rate constants are known, they are difficult to estimate. And so what one is interested is in trying okay? And this is why as they are in general unknown or mostly up to now, most of the story of this is just people just did simulations to understand them, okay, with no structure. So they occur in many papers in systems bio. So I started reading papers in biochemistry, and the first thing I learned is that the meat for us is never there. It's never ever in the paper. If you are lucky, it is in the supplementary information, which is referred. But most of them just use this modeling without even saying it. And I even had an experience. I have a student of mine now, she has a postdoc fellowship with a chemist, and he's a very good chemist. He has many papers, he's really open-minded. He works with people in computer science. But for him, so she talked to him about the equations. I said, equations? He had no idea that there were differential equations. For him, this is the output of sim biology in MATLAB, what happening. So he had not even idea of that, okay? And he's a really open-minded chemist. So the equations are there, and they use them, but it's somehow they are hidden. They don't pay attention that these things are there. So most of the time, I will assume mass action kinetics. And the structure theory was developed by chemical engineers. One, in fact, Feinberg is in math department too. But the tools from algebraic geometry are pretty new. I think this was started by Karin Gatterman, who passed away very young. And then we started something, and there's several other people starting popping out with this kind of tools, but this is really in the very last years. I won't read this now, but you can read then online if you want, or I can send it to you. Okay, so let's give another look to this equation. So this derivative, the vector, lies in the span of these vectors yi minus yj. So it is more or less evident, as this derivative is there, in the subspace, I'm going to call S the linear subspace generated by these differences. So it is obvious that the derivative is there, and so we have this subspace S, and so the trajectory is going to lie in a parallel translate of this subspace. So the dynamics is going to happen in affine subspaces, which are parallel translates of this S, and they will depend on the initial condition where you start. Okay, so if this is S, and we will start here, we will move around here, or if S is not drawn here, but if we start in any of these colored triangles, we will stay there for the rest of the time. This is set there. So this where to start depends on the initial condition, and you have a subspace which is translated to a point in the positive orthon and then intersected with the non-negative orthon, and it is easy to see from the form of for this form that I showed you in the Hungarian lemma, that in fact the non-negative orthon is forward invariant for the dynamics. So if you start inside, you can go, some species can go to 0, but they can never become negative. You never leave the non-negative orthon. And just another piece of notation, so there are linear equations that describe S, so the translates, and these linear equations are called the conservation relations. They hold all along the trajectory. So the steady states of the system are the positive, so the points where all derivatives vanish, so they are the positive real zeros of the Fs, and so this is a real algebraic variety. And this multistationarity means that, so we have in this picture, this blue thing is supposed to be the zeros of the Fs. the steady states, and each of these segment is going to be one of these stoichiometric compatibility classes. So if we start here, we can only move here. So if we go to a steady state, this is the only possible one, while here there are three. Okay, it depends on where you are. You have to intersect your variety with these linear spaces, and then if there is more than one point of intersection, we say that there is multistationarity. There's more than one possible steady state for the dynamics. Okay, why is this an important thing? So Delbruck was a biologist, and at that time they already realized, now they know that most of the cells have the information of the whole to produce all the different cells in the body, or in an animal or whatever, a plant. And so they have the same genetic information, but there are differences between you and me and between the cells in the skin or the cells in the eye or the cell here. And they say that this is produced because of multistationarity. The fact that there are different steady states is what produces cell differentiation. So this is a basic and important thing. This is used, this multistationarity used by the cells to produce some decisions. And then there is this important person in the area called James Farrell. He's from Stanford. He had a major in math and physics and then became a chemist and an MD. And he wrote a very nice short note for biologists. He explains that the current state of system biology says, I quote this from his article, is like planetary astronomy science before Kepler and Newton. And he also says that it cannot be studied now without math and physics. And so what happens is that, what this means is that the basic laws are not known. There are no basic laws known for how the biology evolves, okay? And in fact, in general, there are many, the networks are very complex, but most of the time there's a few variables or some special subnetworks that govern the qualitative aspects. And so this is also supported by the observation that even very simple networks can produce very complex dynamics. And I took these two sentences from this article by Kaufman, Soule, and Thomas. Maybe you've heard of Soule. He works in Arakello theory, but he has a few papers in biology, which are very interesting. And so I'm going to speak about multistationarity, essentially, in my talk today. So this is a very, very schematic way of how an enzyme functions. But the idea is that here there are 4 species. Then first species, this S0, which is called the substrate. And then E binds the substrate. There is an intermediate complex, this is reversible. And then there's a catalysis, and we get S1, this is a new complex. And E goes away. So it produces a reaction, and then it goes away. And why would this happen? And so these people, Fischer and Krebs, got a Nobel Prize in '92 because they discovered that what they were doing is adding a phosphate group and taking it out, and this is one of the ways of transmitting energy. Okay. So the idea is that this binds, and then S1 is just S0 with this phosphate added. So many of the reactions there are other metabolic reactions, but these are very important ones. So this is a case of, this is the network corresponding to 2 sequential phosphorylations. So let me try to read it for you. So there are 2 enzymes. One is E is called a kinase, and F is called a phosphatase. The phosphatase takes away the phosphate, and E puts it. So we start with our substrate. E, this reaction is the same as I had before, but then E acts again. There is an intermediate complex, and we get S2. So this is a doubly phosphorylated substrate. It has 2 phosphates. And then F takes this away and away. Okay? And if you see here, there are 9 species. this S0, S1, S2, and the intermediates and the two enzymes. There are 10 complexes. These are we have a graph with 2 connected components, each of them with 5 vertices, so 5 nodes, so there are 10 complexes. And these are differential equations. Here I changed the notation. I'm calling the variables x1 up to x9. And you have to also number the vertices, etc. And it has this kind of shape. I'm not just to show you, these are the constant rates. And what happens is that the stoichiometric subspace has 3 linearly independent conservation relations. So in fact, the dynamics is going to happen in a space of dimension 6. Okay. So out of these 9 equations, only 6 are linearly independent. The other ones are constrained by these linear equations. And these conservation relations are natural for chemists because one amounts to the total conservation of E, the other total conservation of F, and the other total conservation of substrate. They are kind of natural. So now I'm going to put a little bit more tools. So if you look for the 0 sets of F1Fs, they coincide with the 0 set of all the polynomials in the ideal they generate in the polynomial ring over R. This means that we take all linear combinations with polynomial coefficients, and all polynomials of this form have exactly the same zeros as the original Fs. And then we try to understand what happens if this ideal is a binomial ideal. What does it mean? It means the following. So if we have an ideal, we can try to, so these Fs generate the ideal in the sense that any other element is a linear combination of them. But we could have other system of generators of the same ideal. If it happens that one can find a system of generators where the generators are given by binomial is a polynomial with just 2 terms. Okay? So if it happens that you can just find generators with just 2 terms, then we say that the system has toric steady states. And for instance, what we proved is that I show you the system with 2 phosphorylation, there's 2 sequential phosphorylation and dephosphorylation, but you could have imagined that there are n of them. And in fact, there could be many, many phosphorylations. And so we proved that this standard and basic mechanism has toric steady state. And this result that was implicit in these previous papers. And now I will show you some other important networks which have a toric steady state. This is something I did with Mercedes. So, my student. So, just let me show you why one would like to work with ideals and not just with subspaces. So, when I came to this field, I realized that all the papers I read, they were dealing with linear combination of the Fs with real coefficients. Now I will try to show you just a baby example of why adding polynomial coefficients could be useful. Okay? So this is an important system. It's a system in one of the systems that occurs in this paper by Schiner and Feinberg in Science 2010. And they show, I will read it for you in a second, but they show that there is one of the species that shows what they call absolute concentration robustness. This means that if we look at the 0 sets of the steady states, then the dimension is positive, but the coordinate yp is constant, independently of the initial concentration of everything. It depends on the rate constant, but not on the initial concentration. So whatever initial concentration you take, there is a positive dimensional variety, but the value of one of the coordinates is fixed. So they were trying to understand this mechanism. So let me try to read it a little bit for you. What's the meaning of this? So there is this X, which can transform itself to these other ways of being. It binds to something, but it's still kind of like XT and XT. So this can transform into this other species. And when it has the form XT, it can self-phosphorylate. So it can acquire a phosphate here. And this XP binds to Y, which is another species, and what happens, it transmits the phosphate from X to Y. And then when it is like XD or XT, it can dephosphorylate. There it acts as an enzyme and it dephosphorylates Y. And this is, if you wish, is a kind of a simple mechanism. It's very rare for a positive dimensional general variety that one of the coordinates is constant, but this is what's happening here, and they were trying to understand, one would like to understand why, which is the mechanism that nature is using to regulate this. Okay? So what I'm going to show in the next transparency, I put names x1 up to x9 for the species concentration, and x7 will represent the concentration of this YP. So what I did, we put this in a computer and we computed the Gremner basis. A Gremner basis gives another, this is a basic tool in computational algebraic geometry, and it gives you another system of generator of the ideal. And if the Gremner basis is reduced, your ideal can be generated by binomials if and only if what you get are all binomials. And this is the case here, they are all binomials. So this is a huge coefficient that only depends on the rate constant. And as you see, there are only multiplications and additions, so it is positive when all the k's are positive. Times x3, x7 minus all these times x3. So at steady state, this is equal to 0. And if we are at the point where x3 is not 0, then x7 is going to be this number divided by this number is going to be constant. So you see on the nose that it has absolute concentration robustness. But what happens is that in the same article where we deal with this, we prove that if you work with combinations over R, you will never ever get this on the nose. So no combination with real, just constant coefficient will show you this immediately, as is the case here. Okay, so this is another example. So people work with this, what are called the motifs. What the motifs, I guess, were defined by Uri Alon. He's a fantastic scientist, he's a physicist, and he works in system biology. He's in Israel. And he has a book on this, and he coined the name motif. A motif is a small network. In his definition, it's a small network which occurs in nature way more frequently than a random small network would occur. And this is because there has to be some reason, but also the only thing that we can understand or study is small networks. Okay? So but we study the small networks that occur frequently. And so this is a paper by these very interesting people, Elisenda Feliu and Carsten Buff. So Elisenda also is from Spain, and her thesis was on arithmetic K-theory before she turned down to its biological applications. And Carsten Buff is from Copenhagen, Denmark. So they studied the multistationarity in all these basic enzymatic mechanisms. And what can be seen is that only C, D, and this last one don't have toric steady state, but all the other basic mechanisms they do have. So why one would be interested in this? Well, what happens, so I'm hiding a few hypotheses here, but essentially what happens that if the steady states are given by binomials, then the toric state variety can be parametrized by monomials. So there is a monomial parametrization. We have monomials here, and then we have some constant or numbers that depend on the rate constants. And T means T1 up to Td is the dimension of the steady state variety. And what's nice is that we could use this to detect multistationarity. So we're going to call V the matrix with rows V1, Vs is the matrix given these exponents here. Okay, and so let me show you the theorem. So finding multistationarity is finding the number of positive real roots of a polynomial system, and this is very hard. This is non-algorithmic, but it is algorithmic, but it's very hard. But then what happens is if the system has toric steady state for all choice of rate constant, then it becomes easy, because this can be translated into the following. So there are two subspaces. One is the linear subspace S, which is the subspace generated by the yi minus yj. And we have another subspace S, which is the image of this matrix V of exponents. So we have two linear subspaces. A linear subspace will intersect some orthan. It could be of smaller dimension. It could intersect some orthans, or some other ones not intersect. And what happens is that there is multistationarity for some choice of rate constant, if and only if there is a point in the image of V and a point in S which have coordinate-wise the same sign. So if they do both of them, if there is one orthan of any dimension, but not just the origin, where they both intersect, then this is a witness of multistationarity. And this can be checked. Plus more detail that I'm not saying now. So let me just give you a concrete mathematical open problem. So if one considers n sequential phosphorylation, I'll show you the system for two of them. So there's a very nice paper of Lingmi Wan and Eduardo Sontag. He was originally from Argentina, left long ago. So they proved that there cannot be more than two. Stoichiometric compatibility means in this one of these translate of the what's called the stoichiometric subspace S. That cannot be more than 2n minus 1, and this is already an improvement because the obvious bound is 2n plus 1. And then they prove, so we have the positive orthan in constant rate space, in the space of the kij's. So in fact what happens is that there exists a semi-algebraic decomposition of this space, and in each of the cells in the complement, the number of real roots is the same. And what they were able to do is, in some small corner of this orthan in constant rate space, they proved that there were at least n plus 1 positive roots. And in fact, they conjectured that the actual upper bound is n plus 1. This is only known for n equals 1 and 2. That the bound is 3 and it cannot be more. But in general, it's wide open. And something that has not been understood and should is how many of these could be attractors. Because we want steady state we can attract or not, but maybe if they are not attractors, they produce some cycles or something. Okay, but the nature is not, there's no conjecture of the nature of those, and this bound is unknown. Okay, so let me try to, what happened is, look at me for a second. So what I'm going to tell you the next two transparencies, or three. So I went to a meeting last November in Dagstuhl, which is like Oberwolfach, but for computer science. And there were several people working on this, and we realized that we had this statement using science to detect multistationarity, and there were two very interesting guys from Austria doing also some different thing, but also some sign conditions popping out, and there are many, many other. Then we started realizing that this occurred in many instances, in particular of applied mathematics, where one wants to understand number of positive solutions. So we decided to abstract what was happening. And this is what we did. We tried to produce a mathematical theorem that would explain all of them. We hope that this will be posted in a month or so. So we'll see it's a paper with many authors because it started with a discussion in this workshop. So as we are going to be concerned with the positive real solution, we can use real exponents. We don't need to have natural exponents and not even allow integer negative exponents. We can have any exponents. So assume that this is what we call a generalized polynomial map, because our exponents could be just real numbers. So we have a map which depends on this constants k. We have positive numbers here. We have a bunch of equations. These A and J are real numbers, and all of the monomials have these exponents B. So we have two matrices, the matrices of the coefficients and the matrix of the exponents, and generalized polynomial of this shape. And then we are going to say what we are interested in is if we could have more than 1 0 when some linear equations are satisfied. This can be phrased in this way. We need to understand when the map is injected with respect to S, which means that if we take 2 points which lie in S and they are different, then the value of F is different. Okay? F need not be injective, but if you have 2 points whose difference lies in S, then the values, different points, the image by F has to be different. Okay, so we take a point X, the sign vector is what you imagine, but it's coordinate-wise, plus, minus, or 0. And I'm going to write S star, S minus 0. And so the theorem says that the map Fk is injective with respect to S, but this is the question for all values of k, if and only if there is a sign condition which is satisfied. So the sign condition, for instance, this is, we pick S, a subspace, and this sigma of S are all the orphans that S intersects, non-trivially. Just we exclude the origin. If all the orphans where S, a point of S lies, need not be one of the orphans where the kernel or the points in the kernel of B have signs. And there is this, let me read this for you. This is, we take all the points with the same signs as a point in S. We take the image by B and we compute the signs. But you don't need to read this, but it's a sign condition on where some subspaces, which orphan some subspaces intersect. And what happens is that S is finite or linear subspace. This can be checked algorithmically. And this generalizes many theorems. It's a celebrated Birch theorem in statistics. And it corresponds to the case in which A equals B, and then this is obvious. And then there is a famous theorem by Feinberg, or for complex balance equilibria for delta equals 0. I'm not finding now the efficiency, but it also lies in this framework. But several other theories occurring in the literature in applying to applications to electricity or to economics, and many instances are inside this framework. And so what happens when I saw this, I realized that this was giving, for me, the first partial generalization of what's the Descartes rule of signs. I will remind this for you. So the Descartes rule of signs is very basic. It was proposed by Descartes in the 17th century. You have one polynomial with real coefficients. So let me read it for you. He proposed this in an appendix to this Cours method. And then there is no multivariate generalization. We want an easy thing that just considering signs of the coefficients that gives us an idea of how many real roots. Okay? There was only an attempt by Ilya Ittenberg and Marie-Françoise Roy, but they have a lower bound, but they had a conjecture that was disproven, and there's no conjecture in general. And so this is what the Descartes rule says. If you have a real polynomial, then the number of positive real roots, it says something more, but let's talk about this. It's if bounded by the number of sign variations of the order sequence of coefficients. Let me give you an example. Assume that f is this polynomial here. So there is a plus, minus, plus, plus. So we record just the sign, so that we don't know the sign of A0. I just wrote the sign of A0. Plus, minus, plus, plus. So here there's a sign change from, if there are zeros, we don't consider them. So here there is a sign change, and here is another one. There are two, and depending whether this is positive or negative, there could be maybe, if it's negative, there is one more. So at most, 3. The variation of the sign is 3. So such a polynomial, no matter what is the value of A0, can have at most 3 positive real roots. This is a simple example of his theorem. So let me tell you this, what the previous result gives for me this first generalization. So again, we have a system of equations. We have n equations in n variables and r monomials. So the b's give the exponents. So there's a matrix giving the exponents, and there is a matrix giving the coefficients. So we equate these to CIs, to any value C. And the theorem says this, for any choice, the system has at most one positive solution if the following thing happens. So we have these two matrices A and B. So we can take maximal minors here and there. It could happen that this is 0 and this is not, or the other way about, we don't care. But when they are not 0, what we ask is that the product of maximal minors is always positive. Meaning that, for instance, let's say A simple example of this is the following. So it's written in more or less in fancy terms. If the oriented matrices of A and B are equal, then there is at most one positive solution. This means the following. You have these two matrices. If it happens that all the maximal minors in A and B have the same sign, then there cannot be more than positive real root. It's a little bit more general because we allow zeros. But in particular, if the corresponding maximal minors have the same sign, then this bounds that the real zeros could be 0, maybe they are not. This will depend on the C's. For some C's, there could be 0. For some other C's, there could be 1. Okay, so this we extract from the biochemical reaction networks, but this is the explanation of why this generalizes what is happening in one variable. So in one variable, we have a polynomial. So the exponents are all positive. So depending on the sign of a 0, we allow at most one sign change. And so the Descartes rule was telling us that there could be at most one positive real root. But also looking at this, I realized that we had this case hanging around all the time. What's the role of this case? The role of this case somehow is changing a point by any other, these are positive, by any other point with the same signs. And so if it is an assertion on the signs, it does not only predicate on my system, but only on any other system with the same sign. So we can put here any positive constant and the system has to hold for them too. This is why we can only have this thing, this happens for all choice of rate constants. Okay, so let's go back to biology. So I'm going to show you a couple of examples with what are called bifunctional enzymes. Bifunctional enzyme is a molecule that acts putting a phosphate or taking it away, but it's the same molecule. So the goal that is to understand the design of the different mechanisms. And so any element on the ideal generated by the F is called an invariant because it's a polynomial relation that has to be equal to 0 at any steady state of the system. And so we can just take the F's, but they are not interesting. In general, what happens is that one can measure some variables, some of the concentrations are not others, so one would like to extract some invariant that only depends on a chosen group of variables. So this algebraically is elimination of variables. And so we want to, I will give you now a baby example on how the invariant could be used to decide if a model is wrong. For instance, this is a baby example. So in the sequential enzymatic mechanism I show you, it is easy to see that one gets an invariant of this. So S0 and S2 are the concentration of the substrate and the doubly phosphorylated substrate. This is singly phosphorylated. So we don't know K, because it depends on the rate constant, which are now, this is too simple, okay? But what this says is that these points need to lie on a line. So what we do is we run the experiment, we measure the concentration, we plot the points as 0 times 2 comma S1 squared, and if they more or less lie on a line, then the model is correct. Otherwise, we have to change our model. This is a basic use. And so what we did is we studied, so I didn't mention this, but Jeremy Gunawardena, he's now a professor of system biology in Harvard. He started as a mathematician. He made a thesis in algebraic topology with Adams in the UK. And then he moved out of academia. He worked for Hewlett-Packard for many years. And then while working at Hewlett-Packard, he realized that IBM was paying attention to biology, and he decided that he should. And then he left Hewlett-Packard and ended being a professor at Harvard. But he started just in the lab with doing experiments, nothing related to math, until he found this theory in some notes by Feinberg. And he decided that there was some structure, and maybe he could use his math again. And this is one of the persons who started working in this area with more algebraic tools. So what we did is we took this system by Shinner and Feinberg, which in fact represents mechanism in this E. coli bacteria. And we added just a single reaction. We assumed that Y P can be self-phosphorylated. This is why it was this object that was constant. Then what happens is we found invariants which allow us to say that when now there is no absolute concentration of us, it's not true that the value of this species at steady state is constant, but what happens is that it is bounded by something that depends on the rate constant but does not depend on the initial conditions. So we still get a robust bound. And this is just to tell you that if you are lucky, the best thing that you can find in a paper is something like this. This is very lucky. In general, you will have this picture. If you are really, really lucky, you will have this explaining which are the reactions that are really taken into account, and these are the names. And they keep, at beginners, why don't they call them X1, X2, X3? But if the names just express something, you know by the name what is the function that you expect from the species. They are really, now I use the names when I understand a little bit, because this connects to something in your brain that tells you what to expect in principle. Okay, and maybe if you are more lucky, you see it in these terms with a dictionary or something like this, but in this case, we wrote down the equations, we found invariance, and it happened that this bifunctionality, this was an example, this modeling of something happening in mammalian cells, that there were these two proteins that either one or the other was bounded independently, has a robust bound, depending on the sign of the difference of two of the rate constants. Okay, so I think I have a few minutes because I started later, so it's time. So I will just show you what this maximal response. So this is a system that I took from this paper. This is a well-known and important paper. And there are 11 differential equations plus 3 independent conservation relations. It's very similar to these phosphorylations, two phosphorylations sequential that I showed to you, except that when coming back, there is one more intermediate complex in this way and in this way. It's just something is added, but it's very similar to the previous one. And this is notation one finds. So these are the three conservation relations, the way you find them written. So this is the total, this constant, this is the total amount of kinase. And this is the E, the enzyme, and these are the intermediate that have an E inside. And they tend to think that this is the input, because in principle, at the beginning, there are no intermediates, just E, so this depends on the initial concentration of the enzyme. It's decided, so this is assumed to be the input of the system. And then the output of the system is the total concentration of the doubly phosphorylated substrate at steady state. Okay, so they want to understand what happens, so we vary this input, the total amount of enzyme, and we want to understand which is what they call the maximal response, which is the maximum possible value of the doubly phosphorylated substrate at any steady state. So there is an obvious bound, because one of the other concentration relations, this is linear relation, conservation relations, has a sum of everything having an S. In particular, you have the concentration of S2 is equal to some fixed number. This is one of the equations of this linear subspace, but it has all positive coefficients. S2 is here, so the total amount of substrate is a bound. So S2 cannot be smaller than what they call S dot. But this happens all along the trajectories, all along, okay? And what our aim was to find a better bound for the value of S2 at steady state. And what happens is that in the literature, what they do is, they do this when they can find an analytic expression for S2, which does not exist in this case. And also when they assume they have an expression, so in particular have expressed S2 as a function of this input, so in particular there cannot be multistationarity, because you need to write a function. So what our message is that you don't need to find an analytic expression. You can do elimination in the polynomial ring and find a huge polynomial of degree 6 in the x and 4 in the c variable. In fact, the total degree is 8, it's not 10. So we did this computation with Singular. I don't know if Singular is this fantastic program that they developed at Kaiserslautern, where Gerd Martin Groys was very much involved with it, and it is still. So we computed this polynomial. I'm not showing it to you, don't want to see, but it's huge. When you see it, you realize that you will never find it by hand unless you spend several months. But once it's there, we can use it. So what happens is this picture is the following. This is a C, so here my coordinates are here, C, this is the concentration, the total concentration of enzyme, and this is the value of, this is X, is a doubly phosphorylated substrate at steady state. And the picture is this curve like this, and maybe you realize that for some values of C, there are 3 values of X, so there is multistationarity. And this value here is the obvious, what I call the trivial bound, the bound all along the trajectories, and we were able to find this bound. So when I showed this to Ferrell, he happened to be in Buenos Aires a month ago, less than a month ago, he told me, okay, but why are you so complicated? If you look this way, then C is a function of X. This is true, but then what happens is that this is just, if you do multiplications, additions, even taking roots. Even if we only think on the positive real roots, every conclusion that we're going to extract only holds for the non-negative real bonds and even for the complex solutions of the system. And here what is there is just, this is the part with C negative, it goes up. So the bound we have here, but this you cannot see very well, but it's higher. And there is also a negative part. And here there are 2 asymptotes, one at 400 and something and one at minus 27. And if you want to plot the whole picture together, it's almost impossible, because it's very difficult to find this implicit equation. But there are tools just to understand. So what we are doing, we are plotting the curve PCX equals 0, which has to be satisfied at steady state. But we don't want really to plot it. We say that if we use discriminants and resultant methods from elimination, we can say it's going to be here and there and here and there without plotting it, because it's very complicated to make this plot and to have a sense of, if you do it with the wrong scaling, you get nonsense, you don't understand what is happening. And so in this case, we say we can use tools from computational algebraic geometry to do implicit, what they call dose response curves, okay, which are these curves when X is a function of C. Okay, to conclude, let me summarize what I try to transmit to you. First is we can use algebraic geometry to analyze systems, biology models. That linear algebra does a lot of things, but working over the polynomial ring gives more information. But also, if you work in the polynomial ring, as I told to you, just with algebraic computations, everything that we're going to say also will be valid for complex solutions. So then you need to find things that only pertain to positive real solutions, so you need tools from real algebraic geometry. But if we make a symbolic treatment, then we don't need to have the rate constant, which can be in practice impossible, or even in theory, it can be proved in some cases that they cannot be identified, so you cannot really have them. So our idea is if we try to understand the conclusions from the structure of the model, we can see more than just with one simulation. In fact, they don't do one, they do 1,000 millions of simulations. And so our goal is pick one of these models and describe the geography of the rate constant space, so that just to have this huge orthon in rate constant space and make an algebraic subdivision, so in each of the cells we have a different dynamic behavior. But this is at least starting the same number of steady states, but this is really out of reach in general, because so we have the theory, but even for small systems, there are too many variables and we cannot implement it, so we have to be intelligent how to go and how to use the tools when we can only really compute with a few variables. And then something else to be done is to incorporate more sophisticated algebraic geometric tools. So this, I put there deformation theory, because One is studying a small network. Studying a small network means we have the huge network and we set some variables to 0. So we have a varying family and we are in the fiber where some k's are equal to 0 and we want to extrapolate. So what we need is that this information is continuous, or in algebraic terms flat. So we need to understand why we can extrapolate what happens from a sub-network to the whole network. And also do differential elimination to understand more invariance along the trajectory, not just a steady state. And the super goal would be to understand the basic mechanism in nature, but this is absolutely out of reach for the moment. As I was telling you, in theory we have many answers, but in practice they tend to be too complex to be understood or computed. And what I'm sure is that the answer requires a combination of tools from many, many disciplines, of course, including biochemistry, but also many disciplines in mathematics, and I find this fascinating, and this is all. Thank you.
Thank Alicia for sharing with us this magnificent bridge between relative algebra and application to biochemistry. Thank you very much. Is there some questions or remarks?
How good are these bounds? In some cases?
These bounds, the bounds that we found, they're very good. They are sharp.
The biochemists ever look at really big systems with hundreds of species and complexes, or are they mostly concerned with these smaller systems?
Yes, they study the small ones. These are the only ones they can only simulate. Even if you can simulate bigger systems, they cannot deal with two big ones.
How about the behavior as it was in infinity?
The behavior of? Well, this is, in fact, steady state. You never reach a steady state. This is the limit when t goes to infinity. The value of steady state is a limit. And the maximal response is supremum. It's not a maximum. So this is finally, understanding steady state is what happens in the very, very long run. One important question is how quick you get to steady state. But I'm not studying this now.
Okay, we thank again, Alicia.
Thank you.